Hi, welcome. Thank you for joining us here today. I'm Luis Rayado, and I'm the Business Development Manager for NowBias. I'm here with
Émilie Baudin, I'm Business Developer Manager at Clean Cells. We are both part of Clean Biologics Group.
and we're going to present you what Clean Biologics Group as a whole, via NowBias and Clean Cells, can offer you to support your vaccine and viral vector programs and help you take those programs from benchtop to clinical trials. I will start by presenting the activities of NowBias, and Emilie will then present Clean Cells activities and what solutions they can offer you in terms of biosafety testing. So NowBias is a CDMO based in Nantes. We've been operating since 2005. We are a GMP certified facility from the NSM. We have a staff of little over 40 people, and we have a multi-purpose facility with around 1,500 square meters. We have 4 independent GMP manufacturing suites, 3 BSL-2 and 1 BSL-3. Our activities can support your vaccine and viral vector programs through most stages of clinical development. Our focus is mainly on preclinical and phase 1 and phase 2 clinical trials, but we can eventually support you up to phase 2 or phase 3, depending on the needs and the manufacturing scale. We have a broad expertise in process development at all stages of manufacturing for both upstream and downstream processes. Our activities can be developed in the scope of manufacturing several types of drug substances like inactivated or live viral vaccines, viral vectors, and oncolytic viruses. In our GMP manufacturing suites, we can offer you manufacturing in class A and B for start materials such as cells and virus banks. And also we can perform clinical lot manufacturing for drug substance and drug product in both class A and B and class A and C. As far as process development goes, we have 5 independent BSL-2 labs, which allow activities to be performed on several projects in parallel. We use extensively single-use systems to improve efficiency, minimize cleaning, and avoid cross-contamination whenever possible. We have used several cell culture technologies, and we have experience with technologies for both adherent cells and suspension cells for virus production. We have experience with several types of cells like Vero, MRC5, BHK21, EV66, and CAP cells. We can use several types of technologies such as static containers, microcarriers, single-use bioreactors, and fixed bed bioreactors such as the iCELLis technology from PAL. We perform optimization of critical parameters for cell growth and focusing on virus productivity. We can then perform optimization of critical parameters for downstream processes. We have experience with several types of viruses like measles, rubella, polio, MVA, influenza, and NDV. We can use several types of equipment which we have available in our facility, such as homogenizers, TFF systems, single-use filtration, and chromatography systems. And all these activities are focused really on obtaining the highest virus recovery yields possible. To support these activities, we can also do method development for in-process controls for assays such as TCID50, host cell protein, host cell DNA, benzonase, or other specific assays like PCR-based assays. We can then do the process to scale up to 200 liters, and we can then transfer that process for GMP manufacturing. We have 15 years of experience developing and producing both live attenuated vaccines and inactivated vaccines, but our capabilities extend far to other sectors like viral vector production schemes, as those used for gene therapies and oncolytic viruses for cancer treatment. As I mentioned, we have experience in process development from scales as little as 2 liters up to 200 liters in GMP. We can do these activities for investigational medicinal products, for live attenuated vaccines, inactivated viruses, and viral vectors. We have performed various process and analytical method validations for drug substance production for phase 3 clinical development. And we have performed several technology transfers for the processes developed in-house. We have done around 3 process transfer ins and 2 process transfer outs. Here are some images of the equipment that is used on process development and also at the GMP scale. I will then pass on the word to Emilie, who is going to show you what Clean Cells can do to support these programs.
Thanks, Luis, for this information about NowBio's activities and capacities. Now I will focus on Clean Cells. So Clean Cells was founded 20 years ago. We are GMP certified for quality control testing since 2006. Our facility disposes of BSL-2 and BSL-3 labs. Our staff increased the last few years, and we are now more than 100 people. Last year, we are involved in more than 1,200 GMP studies. A study could be only one analysis or more, so it represents more than 5,000 analyses. We propose our services for worldwide company. Almost 20% of our staff is dedicated to R&D. It could be for our own or for customers' needs. For viral vaccine or viral vector manufacturing, I would like to focus on the quality control guidelines. There are different guidelines to follow according to the localization of the clinical trials and of the nature of the samples. For example, the Chapter 523 of the European Pharmacopoeia is dedicated for the cell substrates used as starting materials. Or we can follow the 2616 for the extraneous agent testing. All our analyses are compliant with Pharmacopoeia or were developed according to the ITH Q2R1 recommendation. The objective of the quality control is to be sure that the final product is safe and secure. As I mentioned, the associated quality control depends on the sample nature, MCB, WCB, end of production cells, the therapeutic application, and the localization of the clinical trial. Clean Cells could help you because we have a strong expertise on biosafety analysis. We dispose of all the requested tests. For example, for the identity of cell substrate, we can propose karyotype analysis. We have a cytogeneticist at Clean Cells. We can propose DNA fingerprinting or transgene identity. We could propose harmonized tests in order to be compliant with both European and US pharmacopoeia. At each step of the process, some quality control must be done. If an antiserum is available, we can propose in vitro and in vivo analysis for extraneous agent in cell culture. If not, we dispose of more than 200 contaminant detection by PCR. It could be for human or veterinary purposes. According to your process, we could propose additional tests. For example, if you use trypsin or bovine serum. Clean Cells, Clean Biologics offers a comprehensive set of services that could support vaccine or viral vector production development from the start of the project until the lot release. Thank you for your attention.
Thank you. And do keep in touch. Get in touch with us if you have any questions or if you want to learn more about what we can offer you.
Thank you.